Well, good afternoon and welcome to MedTech Crossroads. Today is Friday, March 12th of 2021. We are almost to the year mark since we first started this journey together. And today, in just a minute, we're going to be joined by Dr. Ing Pottinger from Columbia, who was with us back in May of 2020 and really enlightened us back at that time. After that, it's the pitch party and Q&A as usual. But right before that, we're going to give just a little bit of news. We're going to skip most of the COVID news today because Dr. Pottinger is going to be sharing a lot of information with us today. But we did just learn this a few minutes ago, that Michigan is now opening up vaccination for all eligible adults by April 5th. That means that if you have some underlying condition, you don't have to be past a certain age, and you're going to be eligible after April 5th. So that is really, really exciting. With that, we're not going to cover the data today because we have someone here who can cover it for us better than we can. And that is Dr. Ing Pottinger of Columbia University, formerly with the CDC. She was an inventor of what is still a gold standard HIV test and came on the show in May of 2020 to educate us all about testing at the time. If you want to know more about Dr. Pottinger and the front row role that she's had over the last year, an article was just done in the New Yorker called The Plague Year by Lawrence Wright. Here's a screenshot of that. You can go to the New Yorker. And we are just so pleased to welcome Dr. Pottinger back to the show. It's good to see you again. Oh, you're on mute.
Thanks for having me.
If there's one thing we've learned over the last year, it's the Zoom mute thing. We just love it.
The mute button, I know. Well, thanks for having me back. I'm glad to be back with you.
It's so good to see you again. You've had so much professional exposure to this, and we were just chatting before. Personal one. Are you willing to share your personal anecdote here of the last day?
Oh, yes, sure. In the state of Utah, where I live now, my family, they vaccinated my entire family because my father's over 70, and my mother and I both got our second shot yesterday, and man, did it knock us out. I had a rough, terrible night. My mother too. My father did okay when he got his. My husband did okay, but I felt like I was in an inferno and in the Arctic at the same time. So
that's
I'm
incredible.
glad you're all going to get your shot soon. Just be prepared that the second one's going to hurt a little for some.
Well, we're so thankful that you're willing to be here today after going through that. And one of the things that you educated me on just the other day, and I'm really excited for the audience to hear about it, is like this immune response that we're trying to provoke with the vaccine and how that works depending whether it's a vaccine or whether it's a natural infection. We'll get to that in a minute. But maybe we can pick up where we left off in May of 2020. You came on the show when we were still trying to figure this all out. And one of the big impressions that our whole audience took away was, I can't just trust, I got a COVID test. I can't just trust that one thing. And you started to unpack for us where in the infection cycle it mattered, where you were going to get good data, where you weren't, what kinds of tests, how many tests you need to get. I think you said one test from each of two manufacturers to really start to have any clue. And this was based on your HIV experience in Africa and elsewhere. We would love to have you catch us up over the last year, because now we've got a lot of retrospective look at different types of tests. What can you tell us about the different test types, what they tell us, what they don't tell us, what's been good, what's been bad?
Yeah, sure. So I think everyone's, becoming very familiar with these terms, the RT-PCR test, which of course is the gold standard test. And then you have your antigen test and then an antibody test. That really is the three categories. In the RT-PCR and the antigen test, both tell you about active infection. They're catching you in that early phase, especially the antigen test. And then you have an antibody test. Well, it tells you whether or not you had a previous infection, regardless of your symptoms. And so you can get a lot of information on, especially a population level, about how widespread COVID is, in America, in your state or whatever the population you're testing is. And so they each play an important role. With the RT-PCR test, And that's probably the most common that most people hear about. And it was the one, that CDC started off with developing and handing out to public health labs, their little debacle around that. And, when you go to your doctor's office and you get your nasal or nasopharyngeal swab and they send it to a laboratory, the most common test is the PCR test. And it is the most sensitive, most specific, gold standard. But you can if you're positive, you can test positive on the RT-PCR test, basically 2 days before symptoms and upwards of weeks after your infection. So well past your infectious period. The antigen test, very different. It's a rapid test. And I don't know, how familiar people are with these small cartridges. You put a sample on and it runs, you get lines. It's sort of like a pregnancy test. That test really is about detecting a viral protein, and it happens within the first week. The guidelines for using an antigen test is basically the first week of since symptom onset. And that test has very good use in terms of, allowing you to expand testing to sites that, maybe can't get a sample and sent to a lab. It's very rapid, but its test performance is not nearly as, let's say, as good as a RT-PCR test. And a lot of people will argue with you that, oh, well, it's important because you're really catching people at their peak viremia, most infectious period. But while true, it doesn't catch everybody in that period. And it's really a poor performing test, especially if you're asymptomatic. And there's tons of data out there to show that basically about 30% of asymptomatic people within that first week are picked up with an antigen test. And that number is going to vary by test, but it doesn't improve by much. And then, of course, your antibody test. And this is where I spent a lot of time talking a year ago, was about the antibody test and how poor performing really they were. And this had to do with the FDA. In their original guidance, they allowed all these antibody test makers to flood the market. I don't know if you remember this period.
We remember all those EUAs that were coming out left and right and back and forth and then
This
getting
was
withdrawn.
yeah. So they didn't issue EUAs for the antibody tests originally. So there was like a 4 or 5 week period where they just let everybody sell their tests. And all they had to do was notify the FDA that they were going to sell a test. And they didn't have to submit data on performance. They didn't, right? Like there was no review process in this. And then they realized what an epic mistake they made. And if you want to see it written out, in fact, 2 FDA officials just wrote a piece about it, admitting their error early on in this. And by then, hundreds of tests had flooded the market. And then you had countries like India buying millions of dollars. I mean, we had states here who were independently buying millions of dollars of antibody rapid tests and finding out that they didn't work at all and had to throw them away. Right. And so then the FDA rolled all of that back, took everyone, said, no, you can't sell anything. You have to submit an EUA with verification data. Well, unfortunately, your antibody response really starts to pick up around 7 days post-infection. But on an antibody test, you really don't detect, let's say, everybody who's infected roughly until 3 to 4 weeks post symptom onset. So it takes about a month, for people to make a detectable level of antibody consistently, in a group of people. Some people you might detect a week in, 2 weeks in, but if you're going to really get to the number of true positives of people who had COVID, you have to wait about a month before people make enough antibodies. And so there's a performance issue with that, depending on when you collect your samples. So a lot of people who knew they had COVID went for an antibody test and they found that they were negative. It's because it was too close in time. So
Wow.
And so what most people don't understand the dynamics of, obviously, replication and then your antibody response in the body and how they perform on each of these tests. And I have to say the antibody tests haven't improved dramatically since, May last year. And I still hold true to, if you want an accurate, let's say, seroprevalence result, so the number of people who had COVID in a population, you definitely have to use more than one test, like in an algorithm format, to get at a more accurate answer. And I'll just give you an example. We're doing one of these surveys in one of our countries in Africa, and we're using this Wantai test, Chinese made, approved and given to us by WHO. And this test in this population is saying that 50% of the population is COVID positive. There's no way. There's just no way that that's true. So luckily we have a second test in there, but you know, and it's coming up with about a 14% prevalence, but you can see the difference. Same population, same blood sample, and yet so wildly different.
Wildly different. So the reliability of those tests still after a year has not necessarily come up past where we were.
Right.
Oh, go ahead, please.
I was just going to say, so when people read these results that will say, oh, in India, you know, they had a 56% prevalence, or in Brazil, in Manaus, it was 72%. You should always take that with a grain of salt. The first thing you should ask yourself is, what test did you use? Like, if you're looking at the data, you're curious, what test did you use? And really look to see if they used two tests. And then understand a little bit more about those test sensitivities and specificities. Because all of those numbers are wrong on some level.
It's always getting closer to a truth value, but then the question is, how close are we to that truth value with what we're looking at? I know that I want to go into the test types a little bit deeper. There was something that has often been asked. I don't know if you have any insight into this, which is, when we see test numbers reported, do you know if municipalities and states and the feds are doing a good job of separating out individuals, or is it pretty much just, when we report cases, we don't know how many individuals were tested? So somebody who was tested 5 times or somebody who was tested 1 time. Do we have any clarity on that?
No, so we don't have what we call, let's say, a case report system in this country that tracks individuals, because you could go to your corner, you know, clinic and then decide you want to go test somewhere else. They're going to count that as total number of tests performed, and not that Eugene had 3 tests or whatever done. So they don't do it by person, just by tests.
Well, and as so I told,
so you can see then how, like, if you might be positive and you get one answer, and this is true for other diseases too, like HIV, when people test multiple times because they don't trust the result from the clinic or whatever. So we can count you know, one person over and over again.
Yeah, that makes a lot of sense. I think we just had a video freeze over here, which maybe we can remedy, but if you can still hear my voice, we'll keep going. And as our audience, as I told them, when our family ended up with what was very thankfully a mild case of COVID, which is not everyone's lot, we went out and did a bunch of tests, which I had the privilege to do. And my first one, which was an antigen, came back negative, but given the symptoms, it was like, we're not so sure. 3 PCRs came back positive, but just a couple days later, a saliva-based PCR came back negative, and there's really no reason it should have been. So I think what I heard you say is that antigen is definitely not as reliable as we'd like to see it, except if you're right in the middle of it, and then the question is, will it still give you a positive? What about the sample prep methods for PCR? Can you say anything about that while we try to fix our video over here?
Yeah, so there, you know, obviously there's tons. You can see when we first started diagnosing COVID, it was a nasal pharyngeal, was that swab that went to the back of basically giving your brain a tickle. And then through various studies, they switched the type of swab and media to do, you know, you could just do a nasal swab, and then it became an oral swab as well. Now they all have different performance characteristics. So same person, if you did a nasal pharyngeal, nasal and oral swab, may give you different results. And that has to do, well, if you're doing an oral swab, it's your saliva, right? It's very different. It comes up a little bit later. It's going to be a concentration difference between your nose where you get infected and replication happens. So it's going to be different. The sensitivity there is not as high, and it catches it at a slightly different time point. So the oral, people use it, but it's not the preferred. I'm going to go on a limb here and say it's not preferred. You can do it because it's convenient, but I think equally convenient is a nasal swab, which across all of the Africa studies that we do, we generally use a nasal swab. It can be self-collected, and its performance is similar to the nasal pharyngeal, which is still the gold standard.
So over the course of the last year, we learned a year ago, make sure we get multiple tests before you declare anything. My own experience was like, oh, yep, that's exactly what Dr. Pottinger said as we went
Mhm.
through that. The antigens still not 100%. You got to be right on top of it if you're going to get a positive.
Mhm.
PCR gold standard, but sample prep matters, and we should consider what type of sample prep it was and whether it was the ideal one. Even there, it sounds like you're still saying multiple tests is better than one.
The RT-PCR test is actually pretty good. I wouldn't go
Okay.
out of my way to get a second test there, but certainly for the other ones, I would like an antibody test for sure, not one. Now the antigen test, the confirmatory test there would be an RT-PCR test. So, if especially if let's say you were exposed to someone, you have suspicions you might be positive, and you took an antigen test and you were negative, then you would go and have a PCR test because it's probably wrong.
And that's the one we're using to confirm with. And then the antibody test, you're saying not going to come up for a while. So even if you're pretty sure you had it, if it comes up negative, it's probably just not long enough.
Well, yeah, and the antibody test in truth has no clinical relevance to you. It's, a lot of people were offering it early on. It was ridiculous, right? You could go to your dentist and he would charge you $300 for an antibody test. But there's no, if you've had it, right? If you're testing positive on an antibody test, you don't really need to, you've cleared your infection by then. So it doesn't have any significance. And if you're positive on it, it's been a few weeks, you probably don't need to quarantine, right? So there's no clinical significance. The antibody test really about population surveillance and understanding, how many people in America or Brazil or whatever, a state had COVID at any point in time. And even that number. So I told you it ramps up about 3-4 weeks. It also, we're learning wanes too. So, but that data is a little less clear. We're a year into it now, and it's been really mixed. Some people lose their antibody response, and will test negative, a year out. That's not surprising. And some people will maintain a higher level of antibodies.
Okay, so this isn't really a strict question of is it going to drop after 3 months or not? It's more of a question of for you, is it going to last or is it not going to last?
Yeah, and it all has to do also with your disease severity. Like some people who had really severe disease tend to have a very strong immune response, and so their antibody levels might stay elevated for longer than someone who was asymptomatic, had very low viral load, whatever. Their immune system wasn't challenged in the same way, and so their antibody levels never really developed to high levels and then wane after a short period of time.
And that's we're going to get to that, I think, towards the end, this whole question of vaccine-acquired immunity versus disease. And I think it's really fascinating for the audience to hear. I guess before we go there, though, let's talk a little bit about transmission vectors, because this is something that's gone all around and everything. And we've come to this understanding of, what masks are there for is to prevent droplet spread. And so that makes sense. There's been this suspicion about enclosed spaces. I know I asked you the question. I said, I think I'm seeing this thing with HVAC systems, and I think HVAC systems are spreading it. And you said, well, I don't know if it's the HVAC systems. It seems like maybe it's more the enclosed spaces themselves and the lack of circulation. But what can you tell us about transmission vectors that we've learned over the last year?
Yeah, it's what really transmits this virus is close contact, is you and me standing, within a few feet of each other and talking. And just regular talking, we produce aerosols, and aerosols of different sizes. So you can have aerosols, right? We're gonna have bigger droplets, and then we're gonna have small droplets. And it's really the big ones that are going to give you COVID if I have it, because it has enough virus particles in there. So the concentration of virus is high enough for me to infect you. Now the little ones will obviously stay in the air longer and they won't drop. The big ones will drop after about 30 minutes, an hour. But it is the big droplets, the close contact. There's no question that is what spreads the virus. It's not, it's if we were standing 12 feet apart in a ventilated room, it's even if I'm super infectious, the chance of you getting infected is pretty low.
It's really interesting that you say it that way, because I think we sort of intuitively knew that. I'm not sure we've always put that together because we don't know how much it takes to get you infected. But I think what you're saying is it's pretty clear it takes more than just that one passing
tiny little droplet,
tiny aerosol that was there. Dr. Pottinger, I'm going to try to bring up the covidtracking.com curves, because you made a
Mhm.
really interesting point to me recently about tests and cases and hospitalizations when I was asking you about the question of, did we really see a Thanksgiving and Christmas spike? And you sort of changed my mind on that one when I said, I didn't really see one looking at hospitalizations. And you said, well, hold on, look at the other part of the process. And in fact, here we have those curves with the sort of smudge happening across daily cases during the November to, I don't have the dates there, but I think we're sort of November, there's Thanksgiving right there. And it just sort of smudges across, whereas this in hospitalizations made a nice peak.
Mhm.
This, I think you had indicated, probably speaks towards demographics of who's getting infected. So the fact that we didn't see um, a dramatic uptick right here in hospitalizations doesn't mean that people weren't smudging it out by contact, that the curve wasn't getting pushed across. What do you make of the demographics of this? Who's getting infected and when? Clearly,
Right.
getting people together in groups, that's increasing the infections, but
So
who's getting infected?
right. So if you look at those graphs again, actually,
We'll bring them back up here.
yeah, that'd be great. A key point to it is that the hospitalizations are always delayed from the the number of cases, right? People don't enter the hospital right away when they get sick. And so and then they stay in the hospital for a long period of time. So we have this shift that always happens with the hospitalized cases. And that's why they don't perfectly overlap in their time. And we know now, compared to the start of the epidemic, which is that people do better. We weren't keeping people in the hospital long enough, and so people are now staying 2 to 3 months in a hospital, and that is reflective of these curves. And so the death curve and the hospitalization curves always come down, delayed a few weeks compared to the drop in daily cases. And so, I just want to make that clear first as to why
Mm,
they look different. And then, in the early days, if you recall, like during New York, their epidemic, it was really hammering a different population because we didn't have a lot of understanding about who the most vulnerable were, and the mitigation, mass social distancing, people weren't using them as widely. And so our most vulnerable population, our nursing homes, were really getting
mm.
infected in that, especially first and second wave. And by,
And this, of course, is the
summertime,
north, just to let people know, this is the northeast curves that you were just referring to,
right.
northeast region. Yep.
And then by summertime, and as we went to fall, I think it became very clear, especially, I'm sure everyone on this call knows someone who is at high risk, or their grandparents started to change their behavior, frankly, right? Going out dropped dramatically for older people. They were taking less risks, using masks, all these things. And so by the time we got to basically Thanksgiving and Christmas, people were traveling to see their loved ones, and a lot of younger people were traveling. And so just the sheer number uh, obviously was higher in this winter peak, but it was a shift to, as a lot of younger people were getting infected compared to the first two waves. Very so the hospitalizations started to include younger people as well, because more people were getting infected, right? And even in younger population, and younger, let's just say, is under 50, they still have comorbidities, other issues that would send someone to the hospital. So just by the sheer fact that more of that group was being infected, more of them were entering the hospital. And so our demographics shifted very much between the first, second wave to our winter wave, with a lot more younger people being infected.
That's an interesting shift, yeah, to have seen. Appreciate your interpreting that for us. So being in close proximity, that's the thing. And of course, being
That's it.
inside is going to do that. It's going to just kind of keep you closer together.
Mhm.
Um, do you want to make any prognostications about what's going to happen? Do you see, like, when we talked about how, like, Texas, people go
Mhm.
inside in the summertime, and last summer we saw a spring and summer spike there this coming fall. Do you have any, maybe this is more appropriate for the end of our discussion, but do you have any prognostications in terms of what you see happening? Do you think we're going to have enough exposures slash vaccinations to damp that down significantly at this point?
So we're in a rat race with the virus right now,
Mm-hmm.
right? It's kind of whack-a-mole-ish, in that we are trying to get everyone vaccinated, right? This is our tool, and it works, and it's super effective. But we're fighting time because of the variants. And every day you read a new variant story about whether or not the vaccine is effective against it or not. I would caution everyone to look at that data more closely, which I'm happy to get into, but it is a race. And our numbers, if you look at our current numbers, we are at our summer peak. And if you go back to our summer peak, we were alarmed at how high that was, right? 65, 70,000 new infections. That's kind of where we are right now. But we're thrilled about it because our peak was so much higher this time. But I want to remind everyone, we're actually at our summer peak. And it wasn't great then, and still kind of not great now. So, with these states opening up and relaxing social distancing and mask use, especially indoors, I mean, that's cutting our nose off to spite our face, unfortunately, right? So we need to get people vaccinated. We need to support each other to get vaccinated. And we really need to support individuals who might be hesitant. And vaccine hesitancy shown all across the world. Misinformation reaches everywhere. So we have to overcome this, especially in certain groups, and get people vaccinated. It is our only way out of this, right? Because we're showing that the monoclonal antibodies that was first developed to treat severe cases doesn't work against the variants, the Brazil and South Africa variants. Completely don't work at all for some of them. So we're losing tools. And the biggest one we have is vaccines. And people really, really need to understand that is the way we all get out of this. Like, we can't expect everyone else to get vaccinated, and we don't have to, and for it to get better. So go ahead.
One thing that you told me recently, and I thought it was very, very interesting, because I don't think people get this. I know I didn't. I've been staring at it for a year, although the domain of virology is not my expertise. And we actually had somebody come on the show and say, who is well respected, say a vaccine can only do what the infection can do. And I know you heard that and you said, no, that's not true. Help us understand the difference between vaccine-induced immunity and infection-induced immunity. You told me these are not necessarily the same things, and you're advocating for vaccine-induced immunity. Help us understand why they're not.
So if, like I said earlier, let's say you are, especially if you're asymptomatic, you're not going to build up a very strong immune response, and your antibody levels are lower, they can wane faster. And more importantly, let's call it the wild type virus, which most people were infected with, is not effective against these variants that are spreading. So that means you're still at risk of getting reinfected with a variant. And, for people who don't think it's coming, it's here and it's spreading, right? And at 70, 65,000 new infections a day, it's spreading very well. And it will become dominant because the mutations that are happening in the virus, it's an antigenic shift now. The virus has decided the pressure we put on it from all these people being infected and the vaccine, it's figured out what mutations it needs to keep transmitting. And a key one, you'll see these in the papers. The most important infection or mutation is this E484K mutation, and it causes basically the virus to escape your immune response. And so this is why it's so important, one, for everyone to get vaccinated, even if they were infected already, because some people didn't have that initial very strong immune response. So you can still be infected again. If your antibody levels wane. Now there's, people will guess, oh, well, there's T cell mediated immunity, which is all that's true. It's harder to measure that in a laboratory, but your primary defense are these neutralizing antibodies, and you want a high level of it going into the spring and summer when the variants will start to spread, because some of the data is showing that for well, both Moderna and the Pfizer, the Novavax that will come out, it has a reduced effectiveness against them, but it's still effective. And it varies depending on the study you read, but there's still an added benefit of being vaccinated. So if you already had COVID, you should still definitely get a shot. And some people recommended just one dose. It won't hurt you for more than 24 hours, I hope, to get a second dose either. But it is, don't assume that because you've had it, you're, free from getting infected again. In the face of the variant, many people are basically back to zero. You are prime material for the virus to infect again.
I'm gonna tell a story here, and I want you to tell me if it's halfway a decent analogy, because many times we operate in these kinds of analogies. So if a person is relatively healthy and they are out there and the disease comes to them, but it never really sticks. So they've had it, but their body doesn't mount a strong immune response to it. Yes, they've had it. Technically, they've had it, but they haven't generated any of these responsive T cells, B cells, antibodies, other things like that. When they get that vaccine, the vaccine is designed to stimulate that response. So you're gonna get the response at that point, whereas just a passing exposure to it may not have given you that robust response. Is that a fair way to
yeah, that's fair. And the vaccine really hones in on the most important antibodies to make, right? Because they, especially the mRNAs, they all focus on that spike protein. When you get naturally infected, you're going to make antibodies to the whole virus particle. There's a bunch of other proteins in there that you make antibodies to, every piece of it. But what the vaccine delivers to you is a genetic material, especially the mRNA, Pfizer, Moderna. They deliver a piece of mRNA that will go through its translation process in your arm and basically present to your body the spike protein. And that's the protein you want to make neutralizing antibodies to that will help you fend off an infection. And it's working quite well.
Those of us who don't know about vaccine creation, is there something that is done in the vaccine to not only deliver that specific protein, obviously, but to exacerbate your response to it? To stimulate your body to really make a response to that protein in particular?
Yeah. So first you'll get a high concentration of it presented. And so your body recognizes it as foreign and will mount a very strong response to it. I forgot the second half of the answer.
Is it really just a high concentration? Is that it? And that does it? We just overload it and then you have to respond to it? Or is there something that we do in the vaccine to actually stimulate even more of a response?
They add adjuvants to it that cause the stimulation. And that's pretty common with all vaccines. And there are some companies who started, I believe like Merck, who've given up their vaccine because it didn't stimulate enough. And I think the Sanofi one, they've decided to change their adjuvant to see if they can stimulate a larger response to it because their original, concoction wasn't producing a strong enough immune response. And I have to say, we're all kind of spoiled now because the Moderna Pfizer came out first with 95% effectiveness. And so we have this notion that that's maybe how vaccines should work. That's not normal, right? Our flu vaccines each year, and flu is a little bit different because we're trying to match the circulating strain. On a bad year, it's 20, 30%. On an average year, it might be 40, 50%. And on a great year, it's like 60%. So people really need to keep that in mind as to what other vaccines look like and how we are really lucky to have come out with two highly effective vaccines in the first go. And just to be clear, you and I talked about this, comparing Moderna, Pfizer data versus Johnson & Johnson data and AstraZeneca data. I want to make this very clear to people that, at 95% for Moderna and Pfizer, 72% for Johnson & Johnson. So people are trying to be picky and be like, I'm going to wait, see if I can get my Moderna or Pfizer. I don't want the Johnson & Johnson. All of the vaccines prevented severe disease and death, right? Like that was their key clinical trial outcome. and they were all effective at preventing people from dying from COVID. That is the most important outcome for sure, right? So don't be picky about your vaccine. It will stop you from dying. And two, the vaccine trials occurred at very different times. Moderna and Pfizer were very lucky. They got out in front. Their trials happened before the variants emerged. So none of that data you're seeing had the variants complicating their effectiveness. Now, all of the new studies, Novavax, even the Johnson, are in the face of the UK, the South African, Brazilian variants. And I will say that the UK one is the least worrisome, honestly, for me. Between the South Africa and the Brazilian variants, those are the two we really, really need to be worried about, because they can escape your immune response. The UK one's still very responsive to either natural infection or the vaccines.
Interesting. We've got a bunch of questions coming in here about the variants. Just to do a recap here, we've got: is there a need for vaccination if a person has been infected? You answered that question very nicely with the explanation. And a question from Kieran: how do you see the variants affecting the transmission vectors, since they've been said to be more infectious? We'll get into that. There's another question here about looking at data with the vaccines versus the variants carefully. What should we focus on? Let's actually take that one a little bit later, because I think we want to ask you about data sources. Where, after this, where should we be looking? You and I talked about how covidtracking.com is down. Where should we be looking for data, and where should we be tracking it? We'll get to that in a minute, but let's go back to the variants. You had said to me that variants, some are of concern, some are not of concern. Break that down for us. Not every variant should freak us out, but some we should watch.
Yes, there's a lot of hyperactiveness in reporting every mutation that we see. There's this great website. It's called GISAID, and it is the warehouse for all the sequence data globally. Everyone who's sequencing the virus has entered their data on there. And it's actually kind of brilliant that we are able to share this data and track it. And I will say most of it's, 40% of it's coming from the UK, because that's, they were the ones who really went out of their way to sequence all of their infections early on to identify the UK variant. But it is, picking up, I think we're at almost 800,000 sequences in there, which is amazing. So you can check out that site for
What was the name of the site again for our audience?
Sorry, let me just get the exact site. There's a group called nextstrain.org. You can look at it there. And then the other one is GISAID.org.
GISAID.org.
Oh, GISAID, sorry, .org.
GISAID.org or nextrain.org for sequence data. Okay.
Yeah. And that's basically where everyone stores their data and analyzes it. But so you're seeing a lot in the New York Times and all sorts of places about, there's a New Jersey variant, there's a Detroit variant, there's a Louisiana variant, and there's an LA variant, right? We're really hyper aware now, even though, to be clear, we're not sequencing nearly enough in this country to be tracking any viruses, right? Like CDC started off, they were doing 20 samples per state every 2 weeks. And we don't even know when those samples were collected, right? So it's not, think about how few samples that is, given that we were at, an enormous number of infections a day. That's not visibility at all on what's happening at the molecular level. They've increased it. They've tried to do this consortium, because sequencing data is it's hard. It's a little bit labor intensive, and it's not like a PCR test, but the technology has improved dramatically. And their goal, the last I heard, was 7,000 samples a week. But if you still do the numbers, that's a very small percentage of the number of new infections, right? So we're relying on states to do it. And so states are reporting it. It is, we need to do better on that front. And certainly the Biden administration has put some money behind it to expand this. But the ones we really care about, like I said, that E484K is a key. It is key. If you see that in a mutation or in a strain, that is the one that, through all the in vitro work has shown to be the one responsible for escape. A lot of them, and it's in that receptor binding domain on that spike protein that binds the ACE2 receptor. And it is it's so far the most important consequential mutation. Now,
so it's the one that's the best lock breaker to get out around the things that we're putting in place.
Say that to me again.
It's the best. It's the one that is best at escaping and
Yes.
getting itself out of what we're trying to do to it.
That's exactly right. We're applying this pressure through our antibody response, and that is this escape mutation with a host of others. Now, the UK has about 12.5 mutations. The South African Brazilian one are composed of maybe 24 mutations. And so you can see that it's a huge number of mutations, by the way. Like in HIV, which does mutate a lot faster, coronaviruses have a better proofreading. We call it proofreading mechanism when they replicate their genetic material. But it is, these variants were really shocking to virologists that all of a sudden a new variant came out with like 17 mutations, unheard of. And there's a lot of work being done to show that this probably arose from people who were immunocompromised. So then, right, they're not mounting an immune response and in the body. So one, it's replicating in the body longer, and it's allowing it to make mutations. It's selecting for mutations that are increasing its efficiency at replicating. And then it, is transmitted. And that's, if you're wondering where these mutations came from, that's a likely source of it.
Interesting. Immunocompromised patients.
Yeah. And so you're thinking about like cancer patients, HIV patients, right? Like that they are a very special group that we should handle with care because they have huge population, repercussions if that virus is able to come out. And people were confused at first because they kept detecting positive results in these individuals. But then when they did the sequencing, they realized that it wasn't a reinfection, but it was just evolving. And
so basically the virus had a laboratory to work in because it wasn't getting kicked out and it wasn't killing the host.
Yeah.
It was just having a chance to try things until it found out what it wanted to be next.
Exactly.
That's fascinating. We got some questions coming in here. I want to ask some of these later ones first, and then we'll come back to what should we be looking for and where should we be looking for data. I guess a personal one here, a question around antibody results. My family has received positive, negative, and reactive results. Dr. Pottinger helped to understand the difference, especially between positive and reactive. Have we heard this term before?
Yes. So reactive is usually the term we apply to the test that says, if it's a rapid test, if that line is there, we record it generally as reactive. And then your clinical outcome is positive. So if you got a reactive test, it's positive.
That reactive means positive at that point.
Yeah.
Okay. And of course, like you said earlier, negative doesn't necessarily mean negative with antibodies because maybe it just wasn't long enough.
Right.
Question here from Ken, is there any correlation to say low IgG levels and susceptibility or severity to COVID with IgG levels declining with age? Just wondering if we're looking at that. Does the apparent, I'm not familiar with this, but the decline in age, or decline in IgG levels with age, does that play any role in COVID susceptibility?
Well, so people have looked at different antibody levels. I mean, your IgG in general, yes, goes down, right? And that's your primary antibody response. There's IgM, that's your initial antibody response. Your body just throws IgM at it, and then you develop more specific IgG molecules against the virus. And they are longer lasting than IgM. And that's why, by the way, there are two, the antibody tests can target both IgG and IgM or all Ig antibodies. Now, does the decrease in IgG levels in the older population increase susceptibility? I don't think that that's right. I mean, your overall IgG level is not specific. I'm interpreting this question. It's not specific for COVID, so you're still likely to get infected. But
it wouldn't have been up at the time that you got it, but you're probably going to spike once you get it, and so that's going to be the more significant response at that point.
And we see older people still mounting a pretty high level response to COVID when they do get infected. And that's also true when they get vaccinated, that they are producing high levels of antibody response. So.
Very good. Well, this question came up, and I think it's an important place for us to go through here. And you and I are talking about how the Atlantic's wonderful COVID tracking site is now shut down as of March 7th. I think it'll stay up, but the data collection has stopped. Where do we go for that same level of data today if we want to see the same kinds of things that we got so used to seeing there?
Yeah, so the COVID tracking project was such a great thing. They were able to gather all the data and put it in one place. The CDC data, I know we all are familiar with this conversation of who's collecting what data. And if you recall, there was this whole hoopla because CDC originally collected the data. But at the time, that data wasn't being collected fully. Only 60% of hospitals in the country were reporting. And then they would model out the rest. And the White House task force, particularly Dr. Birx, and because we do this in our African countries, we know you can collect granular data. We know you can collect complete data, and we know you can collect data at all hospitals. There is really no reason that states should be totally missing. Like Northern California, there wasn't a single hospital reporting it. But at the time, we also were dishing out remdesivir and PPE, right? And so there was this little, I don't know, you could say struggle, but I actually know that it wasn't a struggle. HHS took the data reporting into its own hands, into a new system, which caused a lot of people to be upset to say, CDC is the rightful owner and collector of that data. But in fact, just for the record, CDC agreed to the move. And that's a piece of the story that never quite made it out, was that CDC was on board with the shift over to HHS. And by the time Dr. Birx was done with it, almost 100% of hospitals were reporting data. And really the key was saying to them, I can't give you remdesivir if you don't tell me how many beds are there, or how many beds you have free, how many are occupied, what your PPE level is. And so that allowed the hospitals to report, even though there was some growing pains in there, the data is pretty solid right now. And if you go to the Atlantic, one of the authors, there's a paper in there about the data that the COVID tracking data was collecting. One of the collaborators in there wrote this piece, and it's solid, and it goes through all the data and how it was collected and how it was reported. And they go through this little struggle between HHS and CDC as well. So to get that data now, your best place is the CDC website. I don't believe that they update it daily. I think it might be weekly, but it is certainly there. So it's a little bit delayed, but it is your best source for it because they're collecting results now properly.
That's awesome. That's great to hear. That's great to hear. And thank you for the insights there that you've had a chance to see. Obviously, you deserve a break. You've seen so much over the last year.
That's why I'm in Park City now.
Get some skiing, and no, we're so thankful for that. As we come to the end of it, is there anything else that you think that we should know as a group? We are so thankful for your insights, and I hope we're not having this conversation again next year. But if we are, I hope you'll join us again and catch us up, because it's always such a wealth of information. What else should we know as we go into this summer?
Well, one thing I wanted to add about data source, there's a NIH, I'm sure everyone's familiar with PubMed, but in there is a specific lit COVID page. You can go there and it breaks it out by diagnosis and all of the different categories and treatment stuff. So it's called lit COVID. Let me just find. And if you go to the top of PubMed there, I think there's a link as well that is specific COVID data papers that you can search.
PubMed lit COVID, and that's going to be research papers directed at COVID. And that's not a pre-pub site. That's actually a published papers one. Just for the sake of everyone watching, we can pop that up here really quick.
It's a great site, and you can see at the top. Yeah, exactly. So you see general, mechanism, transmission. So they even categorize them for you, and I like to go in and just go through different categories and see what the latest papers are. And it's just, you can do a weekly check-in or whatever if you're dorky like me.
That's why we enjoy it. We just love to get the data, and sound bites don't cut it. We just need these kind of deep dives. Well, this is incredible. We so greatly appreciate your participation, Dr. Pottinger, and wish you and your family the best. Hope you get some R&R in
Yeah,
before
and
the
skiing.
next. Yeah, that's great.
We still have a
That's a
couple more
beautiful
weeks.
place to do it.
Yeah. And it feels like normalcy is coming. It will feel like that certainly for as soon as people get vaccinated. But I hope to impress upon everyone that we still need to do things that are like mask wearing and social distancing around people we don't know, obviously, their status of. But it's certainly looking better. I think the hardest part is behind us. And just one quick thing is when the vaccine makers make the pan coronavirus vaccine, we will win this war. And what I mean by that is a vaccine that can target all the variants and potentially future coronaviruses that might cause disease in humans, and by including other bat viruses as well. And that's in the works. So let's hope that this virus I do believe we can do it for this type of virus, not for flu and not for HIV and other diseases. But this one, we have a high potential of being successful at making a pan coronavirus vaccine that will certainly get us out of this one and maybe prevent the next one. And there is definitely a next one.
It seems like this is the kind of thing we've seen more and more of over the years, and this one just happens to be the biggest that we've seen, but obviously similar things in the past at much smaller scales. And that pan coronavirus vaccine, that would be targeting not just a single protein, but how do they do something like that?
Well, so no, they would target the spike protein, but it would be targeting the different mutations, right, that are showing up in the variants. And now that you can go on that GSAID website and see all of the different permutations of mutations, so we shouldn't be surprised that any of them pop up. And then we have bat coronaviruses, and here's even the more beneficial part. There are 4 other circulating human beta coronaviruses that causes the common cold, and one of them really impacts our children, especially. They would include that as well in this pan coronavirus. So imagine if we could reduce colds that are due to other coronaviruses. So
wow. And
the technology is there.
That's fascinating. I'm noticing there's another comment here, and I also want to open up the audience for questions. I think they've been coming in as we've been talking, but if anyone was waiting till the end to ask a question, now's your chance. Type it in the Q&A box. We'd love to get it to Dr. Pottinger. I do want you to touch on this one more time because I think that maybe there's still a misunderstanding about the point that you made. Somebody said the question of don't be picky with vaccines. Well, wouldn't I want the best one for me and my loved ones? Why would I not wait and push for the best one? I think might be good to address that.
So the best one, like I said, the measurement of best is that all of the vaccines prevented deaths, right? The endpoint there is the same. Now that 72%, 95%, you're comparing apples and oranges because of the different timing, right? The Moderna, I assure you, in recent papers, a group out of David Ho's group out of Columbia that just published it, even the Pfizer group has published it, there's a major reduction in neutralizing antibody levels. And out of that David Ho group that was just published this week, it's a 10 to 12-fold reduction. That's a massive reduction in neutralizing antibody efficiency. So when looking at that number, you're not comparing apples and oranges. There's no better or worse. And I will tell you that the Johnson & Johnson one is the most likely vaccine to get the global community out of COVID. It's a single shot. It doesn't require, deep freezing. It's still 72% is still an amazing vaccine, right? In the face of variants. It is our most hopeful vaccine for the planet to get vaccinated, frankly. One shot, right? And now that they've paired with Merck, we'll be able to produce a high number of doses for the global community. It is the one we should all be backing and hoping, is most successful.
That's good information. We do have another question here. This from Ken Spencer, and I just opened Ken's mic up because Ken's a longtime listener of the show and always has great questions. I'm going to let him ask this one live if you're there, Ken.
Yes. So Dr. Pottinger, thank you once again. for coming on. And my family lives in Park City also. So,
Oh, we're all very smart then.
yeah. So my question is that for the under 16 population, without
Mhm,
for the children, so without a large clinical trial,
mhm.
how will the vaccines be approved? Is there some other mechanism that goes on there?
Nope, so they are
Thanks,
in the
Ken.
midst. Yeah, thank you for that question. They are, Johnson & Johnson is expected to release the under 16 clinical trial data by late summer and hoping to get approval in that time. So they will be the first vaccine to do children. Moderna and Pfizer did include 12 and older in their vaccine trial, but didn't have enough data, didn't have enough participants with outcomes that were statistically significant. So that wasn't included in their approval, but I expect that that will come. And the under 12, and that's a really distinct cutoff, by the way. The data is clear that the 12-year-olds start to behave more like adults in how they transmit and get infected. That data is expected by early next year. So all of the companies are working in the child vaccine arena. It's going to be a question of uptake, I think, and not whether or not a vaccine will be available for them and whether or not parents will think it's necessary.
Great. Thank you for that, Ken. And thank you, Dr. Pottinger. We are just love drinking from the fire hose of data and information. And you've had such a wonderful position to be able to see and affect it all from. So we thank you for all you've done and for being willing to share that wisdom with us.
Thank you so much for having me. I really appreciate it.
It's great to see you. Hope to catch up with you again. Enjoy the skiing while you still can.
Yeah. Take care.
Talk soon. Bye-bye.
Bye.
Well, that is just wonderful information, and it's so cool to be able to see what is happening as we get the chance to look retrospectively. I know we all want to be able to prognosticate when something is happening, and it's one of the things we've learned is you just can't. You just got to look back at some of the data and see what has happened, and we're very thankful for that now. Before we get to our pitch party, I just want to share this with you because the MedHealth Cluster, along with Scale Health, is holding a virtual roundtable on healthcare burnout and how to address it. That is Wednesday, March 17th, next week, coming up. You can register at medhealth.org, so you can get there. Also to mention, we at Into Being, as I've mentioned the last couple of weeks, are looking for an embedded systems developer. So if you know somebody whose expertise is microcontroller firmware and user interface software development and system design, and likes working on electromechanical systems and in the realm of regulatory science as well to get cross-pollinated, we would love to talk to them. With that, we're in month 2 of the pitch party. We don't call it a pitch competition because, as our friend Walt Young told us the week before we started this, and he is a legendary investor in these parts, you can't pick winners and losers. Somebody who runs a business well is very different from somebody who pitches well. And we can have a fun time with it. We can talk about what we like and what we don't like, but pitching and running a business are not the same thing. We always talk about this before every pitch, that the gentleman Fred Smith, who started FedEx, was told by his professor, the concept is interesting and well-formed, but in order to earn better than a C, the idea must be feasible. Well, it was feasible, and the professor wasn't able to judge that from his seat. So MedTech startups can send us their pitch deck, 10 minutes long, areas of problem statement, vision, value proposition, team, milestones, business model, competitors, and their ask to pitchparty@medtechcrossroads.org. The first 3 weeks of the month, we let them pitch one per week for 10 minutes, which we're about to do here. And the last Friday of the month, we let our panel of community judges talk about them, what they liked, what they didn't like, and ultimately tip their hat toward one for a $1,000 prize, which Into Being puts forward for that startup. We don't participate in the judging. We let our community judges do that. And so without further ado, I want to welcome to the show John Wilcox. John, if you're there, you can turn your video on.
Great, Gene. Thank you so much. Can you see my presentation deck?
I can see your presentation. Just go ahead and turn it on there. We are not seeing your video though, so go ahead and turn your video on for us.
Sure thing. Here, let me see. Can you see that
There
now?
he is. And of course, my video has died because we're just having one of those days. But you don't need me here, John. We're going to put 10 minutes on the clock and let John Wilcox from Diatech Diabetes present to us. Take it away, John.
Great. Well, good afternoon, everyone. My name is John Wilcox, and I'm the co-founder and CEO of Diatech Diabetes Inc. And we are a company that is personalizing insulin delivery for patients with diabetes. So on my 9th birthday, I was actually diagnosed with type 1 diabetes. Since then, I've used a medical device called an insulin pump to manage my blood sugar levels. You can see here different types of pumps that allow for patients with diabetes to continuously infuse their life-saving medication insulin into their skin. Over 1 million patients worldwide use insulin pumps, yet this technology is far from perfect. The FDA actually reports that insulin pumps have the highest overall number of malfunctions, injury, and death reports in their medical device database. You can see here, there's a picture of a mom who has to wake up in the middle of the night to just check on if her daughter's insulin pump is even working and if her blood sugar is in range. Problems associated with insulin pump use are primarily related to infusion failures of the insulin, which can come in the form of leaks and blocks of infusing insulin, as well as improper pump insertion. Now, over 60% of pump users, or excuse me, over 80% of pump users experience these types of infusion failures. And this puts them at risk for increased costs related to poor diabetes management, hospitalization, and even fatal blood glucose levels. There's a major need for technology that helps adult and pediatric patients, as well as caregivers, better monitor insulin infusion and avoid the terrible health impacts that come with insulin infusion failure. And that is why we are building SmartFusion. This is an artificial intelligence powered infusion monitoring software for insulin pumps. SmartFusion software is licensed and is downloaded by a manufacturer into their specific pump model, and it integrates its algorithms to monitor the pump's data to determine if a patient's infusion is successful. Now, a SmartFusion enabled insulin pump could allow patients and caregivers, such as like a school nurse or a babysitter, to know exactly when infusions fail to avoid the life-threatening blood sugar levels that come with failure. Alarms could be sent to a paired device like a phone or a smartwatch you see on the screen for instant notification to allow patients to act before a blood sugar level skyrockets because of a failure. Also, during checkups, healthcare providers could use the data SmartFusion collects to review a patient's infusion site tissue health and also identify best therapy practices to manage a patient's infusion failure. Now, insulin pumps only detect occlusions in infusion tubing after there's a mechanical failure. Failure alarms can take up to 24 hours. Because of this, only 26% of patients are alarmed by their pumps and their insulin is not getting into them. The SmartFusion is going to be the first software of its kind to instantly alert to common infusion failures, as well as provide crucial data on insulin infusion performance to improve diabetes management. Our initial preclinical testing has demonstrated that SmartFusion's ability to detect at an extremely high accuracy these infusion failures that insulin pumps on the market today can't even detect at all. And we believe that patients stand to gain the most from SmartFusion, yet for licensing insulin pump manufacturers, SmartFusion could help acquire patients dealing with infusion failure from competitors' insulin pump models, and also retain current patients who would have previously left due to issues with infusion failure. Now, patients with diabetes adopt and switch their insulin pump of choice based on which features are offered, which incentivizes manufacturers to provide insulin pump systems with the best feature set. An insulin pump enabled with SmartFusion could greatly influence a patient to choose that specific pump model for their diabetes therapy. Now, for a company like Medtronic, who leads the market, having Smart Fusion as a sort of Intel Inside like technology could help them maintain dominance within a diversifying market by acquiring new patients and retaining the ones they already have. Now, each insulin pump user is worth approximately $10,000 for a manufacturer during a 3-year warranty period. Through a customer acquisition retention model powered by Smart Fusion, companies could generate additional revenue based on advertising and selling pumps with improved infusion monitoring from Smart Fusion. Diatech would make money via a licensing agreement set with the licensing manufacturer, who would pay out like a percentage of revenue to Diatech via an upfront cost and royalties based on agreed KPIs. Any exclusivity would drive a higher payment for a licensing agreement made between us and whoever we're signing with the manufacturer. Now, our target entry market is the insulin pump market, which is growing internationally and domestically with a large focus in digital health and software solutions. Yet, once proof of concept is established within the diabetes space, we believe there's a larger opportunity to apply Smart Fusion to other infusion markets such as immunotherapy, Parkinson's, as well as cancer therapy. At this time, we have filed for US and international patent protection of Smart Fusion with claims covering Smart Fusion specifications and use within the diabetes infusion market products, as well as any other device that's administering medication subcutaneously. Our current regulatory pathway will involve a 510(k) submission, which is going to be performed by the licensing manufacturer using technical assistance from us at Diatech. This will be done to ensure that the FDA approval can be based on indications of use specific to the manufacturer's pump model that is licensing Smart Fusion. Now, our team is highly experienced in clinical diabetes work, medical device software development, and most importantly, half of our leadership has a direct personal connection to diabetes. So it's a personal journey for us. And despite the pandemic, our team has proceeded to raise a pre-seed fund, as well as apply for a $300,000 SBIR phase 1 grant, perform one of the largest customer discovery studies on infusion failure, which has recently been accepted to present at this year's American Diabetes Association conference. So we're very excited about that. But most importantly, we've been able to develop out the alpha version of Smart Fusion. Our milestones ahead involve us completing our preclinical testing, as well as additional legal filing, hiring on more talent, and completing any further clinical validation testing and regulatory clearance required by the manufacturer. So we're very excited to be backed by some of the industry's largest organizations, including the Juvenile Diabetes Research Foundation, and as well as Stanford University. So we're moving fast now to close a $1.3 million seed fund that would allow for 24 testing and company growth that is needed to take us all the way to market entry via a license agreement with that insulin pump manufacturer. And we believe that we would be able to do this by late 2022. We plan to utilize non-dilutive funding from organizations like the NIH, as well as private capital from angel investors and high net worth individuals who are personally connected to the diabetes space to fill out our raise. Now, the truth is that patients are unaware if they are getting their life-saving medication. With Smart Fusion, we are going to change that by making diabetes infusion safer. Thank you so much.
Well, thank you, John. We appreciate that. One of the hardest things for me as we have these pitches on, we agreed that we weren't going to ask specific questions afterwards, and we let the judges judge just on the basis of the 10-minute pitch. So I always want to sit here and just ask people questions. But the one I do get to ask, and you touched on this at the beginning, is tell us just a little bit more about your passion, why it is that you're doing what you do.
Yes, of course. And Gene, do you want me to stop sharing the video so I can just...
That's fine. You can go full screen there and stop sharing the video.
Great. Yes, Gene, that's a great question. So we all have a passion for diabetes on our team. But of course, my own personal one stems from, again, my diagnosis when I was about 15 years ago on my 9th birthday. And since then, I've been very passionate about working with diabetes care and technology. I went through my educational background in college, studying biology and working with a pediatric endocrinology clinic. And that was where I met the other members of our team, where we began solving this problem of infusion failures with an earlier version of Smart Fusion. But since then, we've grown the company to where it is today. And my passion is only bolstered by actually getting to interact with patients and clinicians. It's a fantastic field, but one that needs a lot of focus on making the technology better, because it's a very technical field as well.
Well, we're thankful for what you're doing and thankful you're with us here today. I hope you'll tune in the last Friday of the month when our judges have all sorts of comments. And we greatly appreciate you being here today, John.
Thank you, Gene.
Thanks again. Well, and with that, I want to turn to our audience and see if there's any last comments or questions or announcements. You know that we love to tell people about events that are happening in the local area and get them boosted just a little bit more. So if you have anything like that coming up in the next few weeks, let us know about it. We'll give about 20 seconds for that before we close. We went a little bit beyond today. We usually try to close down by 3 o'clock, but Dr. Pottinger had a lot of cool stuff to tell us, so we went a little bit over. I am looking at the chat and I am looking at the Q&A and not seeing anything more right at the moment. We're going to shut it down for this, the 12th day of March, episode 49, and we're looking forward to episode 50. It's basically the year anniversary in these next 2 weeks. So thank you for being with us. We'll talk to you soon. Bye-bye.